This is Dr. Malathi from Life Sciences Department. This session we will be reading about Watson and Crick model. So the three-dimensional structure of DNA, which was first proposed by James Watson and Francis Crick in 1953. This structure is the widely accepted structure and widely accepted model, which will be having different conformations and different properties. It consists of two helical strands that are coiled around a common axis to form a double helix. So each DNA molecule is comprised of two strands which are coiled around the perpendicular axis. The two DNA strands are called polynucleotides as they are made up of simple monomeric units called nucleotides. So the nucleotides included in the DNA, which includes adenine, cytosine, thymine and guanine. And the ribosugar which is present in the DNA structure is deoxyribosugar. So we call this as deoxyribonucleotides. The two strands are held together by hydrogen bonds and are complementary to each other. The base adenine always interacts with thymine, so A will be interacting with T on the opposite strand via two hydrogen bonds, and cytosine always interacts with guanine, C, which is interacting always with G via three hydrogen bonds on the opposite strand. So always a purine will be interacting with a pyrimidine and a pyrimidine will be interacting with a purine. So never purine will interact because of the space constraint, and never pyrimidine will interact because the interaction is not possible. Bond formation are also not possible because of the distance which is kept between the two strands. So each strand has 5' end and 3' end. So in 5' end, the free phosphate group will be seen. In case of 3' end, free hydroxyl group will be seen. So this will be the general structure of Watson and Crick model, wherein both these strands are anti-parallel in nature. So one strand moves from 5' to 3', the other strand runs from 5' to 3' direction. So both are anti-parallel in nature. At the backbone of each strand are sugar-phosphate backbones. So sugar and the phosphate group forms the entire backbone structure. So inside the backbone are the nitrogenous bases. So all the C, A, T and all will be buried inside and between the two strands. So which will look like ladder. So because of this alpha helical conformation, we have major groove as well as minor groove. And between each of the nucleotide, we have around 0.34 nanometer. And between the strands, we have around 2 nanometer in diameter. And one pitch includes 3.4 nanometer, and each pitch will have around 10 nucleotide in the structure. The strands are anti-parallel, meaning that one strand will run from 5' to 3', the other strand will run from 3' to 5' direction. So already we have studied about the diameter of the double helix, which is around 2 nanometer, and the double helical structure repeats at an interval of 3.4 nanometer, which corresponds to around 10 bases. That's why one pitch carries around 10 base pairs. The deoxyribonucleotide are linked together by 3', 5' phosphodiester bond. It's a covalent bond. This covalent bond will make the particular DNA nucleotide, each strand, to be very stronger. Whereas hydrogen bonds are weaker, the hydrogen bonds are used to hold both the strands each other. Whereas this phosphodiester bond will be used to have the basic polymerization of each of the strand. The structure of DNA is a double helical structure, but it looks like a twisted ladder. It's a helical structure but with a twist. Because of that only we get both major and minor grooves. The side of the ladder, which is backbone, which is made up of sugar, which is deoxyribosugar, along with phosphate group. The middle one will have the ladder's middle portion, which is made up of nitrogenous base. So already we have seen A, T and GC. As a result of double helical nature of DNA, the molecule has two asymmetric grooves. One groove is smaller than the other. The larger groove is called as major groove, occurs when the backbones are far away apart. While the smaller ones is called as minor groove and occurs when they are closer to each other. So these grooves are very important for the organization of the DNA, so that the DNA can wound around the histone proteins and organize itself in the nucleus of the particular cell. Thank you.